Welcome to the deep dive. We're here to cut through the noise and bring you, well, the most compelling insights from really cutting edge research. And today we're plunging headfirst into one of neuroscience's really persistent mysteries, that intricate dance of sleep and wakefulness. I mean, despite decades of dedicated investigation, our understanding of how the brain actually orchestrates these fundamental states, it just keeps evolving, keeps revealing surprising complexities. So for you, our dedicated listeners in sleep medicine, our mission today is, well, to provide a real shortcut into the latest advancements in neurobiology. We're talking specifically about dopamine, DA, and norepinephrine NE signaling, and, their profound implications for narcolepsy. You're likely already familiar with the foundational concepts, so we're planning to go straight to the cutting edge, hopefully revealing some of those aha moments that these new tools are making possible.
Yeah, it's an absolutely critical area. And narcolepsy, well, it affects roughly 1 in every 2,000 people in the U.S., so it's pretty prevalent and often quite debilitating. It's a REM sleep-related condition, right? Often develops in adolescence. And the hallmark symptoms are pretty well known. That excessive daytime sleepiness, really disrupted nighttime sleep. And for those with type 1, there's cataplexy, that certain, often kind of motion-triggered intrusion of REM sleep muscle paralysis, but during wakefulness.
Right. And the personal and societal toll is just huge, isn't it? Impacting productivity, raising healthcare costs.
Absolutely immense. And what's truly striking, I think, despite that impact, is how much we still have to learn about the exact neural mechanisms behind the disrupted sleep and the cataplexy. So today, our focus is going to be largely on the monoaminergic system. That covers key neurotransmitter pathways like norepinephrine, dopamine, serotonin, histamine, and specifically some groundbreaking new research is really shining a light on the roles of midbrain DA neurons in the ventral tegmental area, the VTA, and brainstem NE neurons in the locus coeruleus, the LC.
Okay, yeah, let's unpack how we got here then. Starting with some of those landmark discoveries. I mean, for many of us, the pivotal moment in understanding narcolepsy really came in the late 1990s, right? With the discovery of orexin.
Orexin or hypocretin, yeah.
Which was identified pretty quickly as just key for maintaining wakefulness.
It was fascinating how swiftly that became a cornerstone finding. So researchers, they developed this mouse model where this peptide was basically eliminated. And what they saw was, well, a really striking replication of human narcolepsy type 1 symptoms.
Like the sleepiness, the fragmented sleep.
Exactly. Profound sleepiness, fragmented sleep, and these sudden bouts of immobility during wakefulness. You just immediately confirm that direct link between human narcolepsy and the loss of those hypothalamic orexin neurons. It was a real game changer.
Definitely a game changer for the what of narcolepsy. But it's worth remembering, like you hinted, that decades of pretty ingenious research had already laid some crucial groundwork. I mean, you mentioned studies going back, 50 years?
Yeah.
Using those naturally occurring canine models. What did those early insights tell us about monoamines? And, I mean, how did they even get that info with the tools back then?
Well, those early canine studies were really remarkable for their time, especially considering the tech limitations. Through really meticulous neuronal recordings and some pretty precise drug manipulations, researchers first started to hint, at the role of what they presumed were DA and NE releasing neurons.
Presumed, right. Not directly measured yet.
Not directly, no. But for instance, they found that pharmacological tweaking of specific dopamine receptors, even doing direct injections into the VTA, could dramatically alter how often cataplexy episodes happened.
Wow, that level of specificity 50 years ago.
It's pretty amazing. And even more compelling, recordings of what they thought were norepinephrine-containing neurons in the brainstem showed something really profound. Their activity was consistently high during normal waking, but then it just plummeted near zero during cataplexy.
Okay.
And maybe the most impactful finding was that when they artificially boosted the amount of norepinephrine binding to brain receptors, cataplexy could be really powerfully suppressed. So these findings, yeah, they weren't direct measurements of the neurotransmitters themselves, but they provided incredibly strong early support that dopamine and norepinephrine systems were, playing a significant role in regulating cataplexy.
Yes.
It really set the stage.
They absolutely set the stage. But, like you said, limitations were there. So, fast forward to today, and we're seeing this, explosion of new tools revolutionizing research. One of the biggest leaps seems to be moving from those complex dog models to genetically engineered mouse models. What's the huge advantage there?
Oh, the advantage is immense, and it really comes down to the genetics. We can now precisely target specific groups of neurons, through selective modification of the mouse genome. This lets researchers manipulate brain circuits with a level of specificity that was just unimaginable before.
Right.
And we have several really robust mouse models of narcolepsy now. They reliably replicate key aspects of the human condition, disrupting the orexin protein or its receptor, or even just getting rid of the orexin neurons themselves. And crucially, these models let us use these advanced neuroscience techniques that have just dramatically increased the resolution for recording and manipulating neural circuits. We can ask entirely new questions now.
Okay, so speaking of those advanced techniques, optogenetics, often hailed as maybe the most impactful neuroscience innovation of the century, right? For
Yeah.
precision manipulation, anyway. For those of us not in the lab day to day, how does this sort of light switch for neurons actually work?
It really is revolutionary. So imagine you deliver a gene, okay? And this gene expresses a light-gated ion channel, but only in specific types of neurons you're interested in.
Okay.
Then, when you shine light of a very precise wavelength onto that brain area, these channels open or close, and you can either activate or suppress activity in only those targeted neurons.
Wow.
It's just a monumental leap over the old pharmacological ways, which might last for hours and just, slosh around the whole brain, affecting
Well,
everything.
more specific, yeah.
Way less specific. Optogenetics gives you exquisite temporal-like, down to milliseconds and spatial specificity. It's like having a remote control for tiny, specific parts of the brain.
So if optogenetics is the remote control, how do we actually listen in on what those neurons are doing in real time? That's where fiber photometry comes in, right? Sounds like the perfect partner.
It absolutely is the perfect partner. Fiber photometry also uses light, but instead of activating or inhibiting, it's used to record their natural endogenous activity. It started out recording general neuron activity like calcium signals, but its real power, I think, emerged when it was adapted to record specific neurotransmitter release.
Oh, okay, that's the key.
That's the key. Using these specialized genetically encoded biosensors that have been developed for dopamine, norepinephrine, and others. So what's genuinely exciting here is the combination. Optogenetics for manipulation, fiber photometry for real-time recording.
Paired with the mouse models.
Exactly. Paired with those robust mouse models of narcolepsy. It lets researchers identify the neural circuits contributing to sleepiness and cataplexy with just unprecedented precision. We're not just inferring anymore. We're actually seeing it happen.
All right, this is where it gets really interesting for me. Because these powerful tools, they're uncovering genuinely novel insights into how dopamine and norepinephrine actually influence sleep and cataplexy. We've known for ages about dopamine's link to reward, positive emotions, especially in the limbic system. But what's the sort of evolving picture here, connecting it to emotion, REM sleep, and cataplexy?
Right, this is a major shift. So think about it. Both REM sleep, where we have vivid dreams, and cataplexy, which is so often triggered by strong emotions, they both have these undeniable emotional components. So a really compelling hypothesis emerged. Maybe dopamine release in the limbic system actually promotes either REM sleep or cataplexy.
Okay, that makes intuitive sense.
And recent findings are profoundly supporting this. Get this.
Yeah.
A pivotal study in 2022 found that dopamine is indeed released into the amygdala, a key emotion hub, immediately before REM sleep starts in healthy mice.
Wow.
And here's the kicker. Just by suppressing that dopamine release right there in the amygdala, they significantly reduced the total amount of REM sleep the mice got.
Just by controlling dopamine in that one spot.
Exactly. And they extended this to narcoleptic mice and found that increasing dopamine release in the amygdala actively promoted cataplexy. Our own lab's research using fiber photometry with a dopamine biosensor has really helped cement this too. We've shown dopamine release in the nucleus accompanies another key limbic region, reward and motivation. It gradually increases during NREM sleep bouts that then transition into REM sleep.
Okay.
And this dopamine release during NREM was also sensitive to REM sleep pressure. So if you deprive mice of REM sleep, you see higher dopamine levels afterwards during NREM.
Interesting.
And crucially, using optogenetics to inhibit that dopamine release directly decreased how often they went into REM sleep. Now, in the narcoleptic mice, we consistently saw dopamine release in the nucleus accumbens spike before cataplexy and stay high throughout the episode.
Right.
And even more remarkably, just artificially increasing dopamine release in that region was enough to make cataplexy happen more often.
So the link seems pretty direct then.
It really does. These findings strongly support the theory that, emotional stimuli might trigger cataplexy by ramping up dopamine release in these critical emotion-regulating brain areas. The sort of general takeaway is becoming clearer. DA activity increases during both REM sleep and cataplexy and seems to actively contribute to these states.
That is a profound shift, moving dopamine beyond just reward into the very mechanics of sleep and these emotional states. Okay, let's pivot then to norepinephrine. Nora has traditionally been seen mainly as a wake-promoting neurotransmitter, right? Keep you alert.
That's the classic view, yeah.
So what surprising nuances are emerging now about its role in REM sleep regulation? It feels like we're constantly reevaluating these classic players.
You're absolutely right. And this is where recent studies have really challenged those traditional views. I mean, yes, IN certainly promotes wakefulness and vigilance, no doubt about that. But new research is showing that norepinephrine is also released during NREM sleep. And maybe surprisingly, it seems to specifically suppress entrances into REM sleep.
Ah, so it acts like a gatekeeper against REM.
Sort of, yeah. Our lab's preliminary work using direct measurement of NE release with fiber photometry.
Yeah.
So seeing the NE itself found that NE release in the midbrain is high during wakefulness and NREM sleep, as you'd expect. But it's significantly suppressed during both REM sleep and cataplexy.
Okay, so drops in both states.
Exactly. And this data really extends those earlier findings from the dog model because we now have direct real-time measurements of NE release, not just inference from neuron firing.
And the spatial specificity you mentioned?
Right. We have the spatial specificity now to start teasing apart which downstream brain regions mediate NE's effect on cataplexy. And in follow-up studies, we used optogenetics to activate those norepinephrine-containing neurons in the brainstem, and doing that powerfully suppressed entrances into cataplexy.
Wow.
So these findings are genuinely exciting. They point toward a very specific kind of nuanced role for midbrain norepinephrine signaling in regulating both REM sleep and cataplexy, with any activity reliably dipping during those particular states.
It's incredible how these fundamental discoveries are directly impacting how we think about existing treatments. Let's connect this to clinical applications. Take sodium oxybate, widely used, very effective for narcolepsy. It's known for its short half-life, usually given twice nightly, right? To promote restful NREM sleep and cut down cataplexy.
Correct.
But there's always been this slightly curious phenomenon associated with it, hasn't there?
Yes, and it raises an important question that's been a bit of a puzzle. While the sustained effect is clearly beneficial, sodium oxybate can actually initially promote both REM sleep and cataplexy shortly after someone takes it.
Right, which seems counterintuitive.
It does, and the mechanisms behind that initial increase and the later decrease were largely unknown. But our recent findings and work from other groups are starting to clarify this. Sodium oxybate is thought to work as a weak GABA-B receptor agonist.
Okay.
And we know that norepinephrine neurons in the locus coeruleus are inhibited by GABA through those GABA-B receptors. And our research now shows robust NE release potently suppresses REM and cataplexy.
So you can see where this is going.
Exactly. A very compelling hypothesis emerges. The possibility is that sodium oxybate initially suppresses NE neuron activity because of the GABA agonism. That promotes restful NREM, but also allows more REM sleep and potentially cataplexy. Then, as the drug starts to wear off, the subsequent rebound of norepinephrine release could kick in, helping to maintain wakefulness and suppress cataplexy later on.
Ah, okay, so it's like a two-phase effect explained by the NE dynamics?
It seems plausible, yeah. It's a fascinating dual effect, and these new insights are helping us unravel that neurobiological dance, moving beyond just knowing that it works to understanding how.
And what about other promising treatments like histamine H3 receptor inverse agonists? How do they fit into this evolving picture of monoamine regulation?
Right, so histamine generally plays a critical role in promoting arousal wakefulness. H3 receptors are interesting. They're inhibitory autoreceptors located in the histamine neurons themselves, meaning they normally act like brakes to decrease histamine release.
Okay, so they turn histamine down.
They do. So if you use an inverse agonist for these H3 receptors, it basically takes the brakes off, resulting in increased histamine release. This, in turn, is thought to stimulate the broader monoaminergic system, including those norepinephrine neurons back in the locus coeruleus.
I see. So more histamine leads to more NE.
That's the likely downstream effect. The net result is probably a significant increase in norepinephrine release, which then promotes wakefulness and, crucially, suppresses cataplexy.
Makes sense.
Now, the specific circuits and the full mechanisms for how these drugs reduce the burden of narcolepsy are definitely still being fully worked out. But these findings give us invaluable insights, a clearer map, really, for developing even more targeted treatments down the line.
So, okay, as we synthesize all this progress, from those foundational discoveries like orexin to these really cutting-edge insights using optogenetics and photometry, What does it all mean for the future of sleep medicine? What significant strides have we really made in understanding the genetics, the neurochemistry, the specific circuits in narcolepsy over, say, the last 50 years?
I mean, we've genuinely come an incredible distance. The critical advancement, I think, provided by these recent studies, especially those directly measuring DA release in the limbic system, mapping NE activity during REM and cataplexy, has profoundly reshaped the field.
Hmm.
These findings are shining a much, much brighter light on the precise circuits through which these monoaminergic nuclei signal to regulate sleepiness and cataplexy.
Moving beyond just knowing the players to knowing the pathways.
Exactly. We're moving from a broad understanding to incredible circuit-level detail. And that's really where true therapeutic breakthroughs tend to happen.
Hmm.
And if we can connect this to the bigger picture, the relevance goes way beyond just narcolepsy. These discoveries are fundamentally informing our understanding of sleep regulation in healthy people too, and also sleep disturbances in other complex conditions, think Parkinson's disease,
Mm.
REM sleep behavior disorder.
Mm-hmm.
And consider this, the very same circuits we're discussing today, they're also deeply involved in attention and cognitive function, which offers a very plausible, neurobiologically grounded explanation for why poor quality sleep so often leads to decreased daytime focus and other cognitive issues. It really is all interconnected.
Yeah, it certainly seems that way. It's clear that while, okay, many questions definitely still remain in the neurobiology of narcolepsy and sleep disorders generally, these findings really do represent a meaningful and significant step forward towards a more complete understanding of the neural circuits governing how we sleep and wake. So maybe a final thought to leave you with. Given this intricate balance we're seeing between dopamine and norepinephrine and this emerging understanding of their really precise roles, sometimes promoting, sometimes suppressing aspects of sleep and wakefulness, how might future therapies for narcolepsy and maybe other sleep disorders move beyond just broad modulation? Could we get to truly personalized circuit-specific treatments? That's definitely something to ponder. Thank you for joining us on this deep dive.